Okay, preheat your cellular cooking device to 375 degrees Fahrenheit. Let's make the phospholipid bilayer. This is the cellular membrane that surrounds a cell, letting some things in and keeping others out, aka regulating what enters and leaves the cell. Many molecules cross the cell membrane via diffusion and osmosis. It gives the cell some structure and holds everything in place. And the membrane has many proteins embedded within it. Let's add the phospholipid bilayer. The membrane is composed mostly of a double layer of phospholipids arranged tail to tail along the length of the cell. The structure is called the lipid bilayer. Make sure your phospholipid bilayer is packed nice and tightly. And then we're going to pop it in the oven to make sure it keeps its shape. Put your cellular cooking device for 5 minutes. And take out your phospholipid bilayer. Let's take a closer look. The head of the phospholipids are polar, as is water, and the tails are nonpolar. The heads interact with water, but the tails are repulsed by them. Since the cell is constantly immersed in water, the lipids form a double layer, with the heads towards the water and the tails inside, away from the water. Let's make the Golgi apparatus. The purpose of the Golgi body is to manufacture and package proteins before shipping them away to parts of the cell. The function is to receive the proteins from the endoplasmic reticulum, then wrap them in a membrane. The whole package is called the vesicle, and then sends them on their way. The Golgi bodies are located throughout the cell. The structure of the Golgi apparatus is that it is composed of stacks of membrane-bound structures known as cisternae. An individual stack is sometimes called a dictyosome. From the Greek, dicton means net and soma means body. A mammalian cell typically contains 40 to 100 stacks. Between 4 and 8 cisternae are usually present in a stack. Each cisternae comprises a flat, membrane-enclosed disc that includes special Golgi enzymes. Don't forget to preheat your cellular cooking device to 375. And let's make the endoplasmic reticulum. The purpose of the endoplasmic reticulum is a series of membranes that carries proteins from the nucleolus to any other place in the cell. The endoplasmic reticulum was designed to carry proteins from the nucleus to the various organelles in the cell. Though some cells can produce their own proteins, such as the mitochondria and chloroplast, they still need instruction from the nucleolus itself. This is where the endoplasmic reticulum comes in. After the nucleolus manufactures the proteins, it sends them along their way to the organelles by way of this membrane. The ribosomes, anchored along the length of the membrane, also produce proteins that the endoplasmic reticulum carries. However, only the rough endoplasmic reticulum contains the ribosomes. The smooth endoplasmic reticulum doesn't. Its function is its functions in several metabolic processes, including synthesis of lipids, metabolism of carbohydrates and calcium concentration, drug detoxification, and attachment of receptors on cell membrane proteins. It is connected to the nuclear envelope, and is found in a variety of cell types, both animal and plants, and it serves different functions in each. The smooth endoplasmic reticulum also contains the enzyme glucose-6-phosphatase, which converts glucose-6-phosphate to glucose, a step in glucogenesis. The smooth ER consists of tubules and vesicles that branch, forming a network. Let's make the cytoplasm. The cytoplasm is a thick liquid residing between the cell membrane holding all organelles, except for the nucleus. All the contents of the cells of prokaryotic organisms, which lack a cell nucleus, are contained within the cytoplasm. Within the cells of a eukaryotic organism, just like we're making today, the contents of the cell nucleus are separated from the cytoplasm and are then called the nucleoplasm. In eukaryotic cells also, the cytoplasm contains organelles such as mitochondria, which are filled with liquid that is kept separate from the rest of the cytoplasm by biological membranes. It is within the cytoplasm that most cellular activities occur, such as many metabolic pathways including glycolysis, and processes such as cell division. The inner granular mass is called the endoplasm, and the outer clear and glassy layer is called the cell cortex, or the ectoplasm. The part of the cytoplasm that is not held within the organelles is called the cytosol. The cytosol is a complex mixture of cytoskeleton, filaments, dissolved molecules, and water that fills much of the volume of a cell. Make the nucleus. First of all, you want to make your nucleolus and put it in the center of your nucleus. The nucleus is the control center of the cell. It contains DNA and is surrounded by a double membrane called the nuclear envelope. It's usually the easiest organelle to view under a microscope, and usually one nucleus per cell. Indicated by the spaces in the sugar coating is the nuclear envelope, which is perforated by pores and is contiguous with the ER. The nucleolus is a non-membranous organelle in the center of the nucleus and is involved in the production of ribosomes. Right now I'm putting the chromatin, which is located in the nucleolus. Chromatin is a material consisting of DNA and proteins. It is visible as individual chromosomes in a dividing cell. The nucleus is what controls the actions inside of the cell. It's usually the brain of the cell. It contains a DNA and RNA, which works together to build and control. It produces proteins, which are carried by the endoplasmic reticulum to the various parts of the cell. Let's arrange our cellular cake monster. First, what we want to do is make the nucleus contiguous with the endoplasmic reticulum. So take your endoplasmic reticulum and surround your nucleus with it. 2 to 3 pieces should be good enough. Next, you want to make your smooth endoplasmic reticulum and put it on the other side of the cell. Next, take your stack of pancakes, aka your Golgi apparatus, and make it contiguous with a rough endoplasmic reticulum. Next, you want to add your mitochondria. Notice the folded inner membrane in the inside. This is called the cristae. The mitochondria is the energy factory of the cell. This double membrane organelle takes glucose and combines it with oxygen to produce carbon dioxide, water, and ATP, which is a chemical energy usable by the cell. The cell usually contains several mitochondria, and interesting to note is this organelle is almost a cell in itself. It has a double membrane, its own DNA, and can reproduce by splitting itself. Let's make the cytoskeleton. Cytoskeleton is both a muscle and a skeleton and is responsible for cell movement, cytokinesis, and the organization of the organelles within the cell. It is unique to eukaryotic cells. It is a dynamic three-dimensional structure that fills the cytoplasm. This structure acts as both muscle and skeleton for movement and stability. The long fibers of the cytoskeleton are polymers of subunits. The primary types of fibers comprising the cytoskeleton are microfilaments, microtubules, and intermediate filaments. Now let's add our peroxisomes, which are organelles that contain oxidative enzymes such as D-amino acid oxidase, urate oxidase, and catalase. They may resemble lysosome, however they are not formed in the Golgi complex. The function is to rid the body of toxic substances like hydrogen peroxide or other metabolites. Lysosomes are the cell's garbage disposal system with 3 functions: to degrade the products of ingestion, to degrade worn-out organelles such as mitochondria, and to handle the products of receptor-mediated endocytosis, such as the receptor, ligand, and associated membrane. Lysosomes and peroxisomes are located throughout the cell anywhere. Make sure you put a lot of lysosomes next to the Golgi apparatus. Now let's add some ribosomes to the cell. Ribosomes are organelles that consist of RNA and proteins. They are responsible for assembling the proteins in the cell depending on the protein production level. Ribosomes may number in the millions. They're typically composed of 2 subunits, a large subunit and a small subunit. Ribosomal subunits are synthesized by the nucleolus. These 2 units join together when the ribosome attaches to messenger RNA to produce a protein in the cytoplasm. There are 2 places that ribosomes usually exist in the cell, suspended in the cytosol and bound to the endoplasmic reticulum. These ribosomes are called free ribosomes and bound ribosomes respectively. In both cases, the ribosomes usually form aggregates called polysomes. Free ribosomes usually make proteins that will function in the cytosol, while bound ribosomes usually make proteins that are exported or included in the cell's membranes. Our cell is finished. Eukaryotes, just like you and me, are made of millions and billions and trillions of eukaryotic cells. Bon appetit!